Hello, ladies and gentlemen. This is Dan Montano, Chief Executive Officer of Angiogenesis Medicine. I want to welcome you to our webinar on Can Therapeutic Angiogenesis Extend Your Life an additional 30 quality years? Dr. Jacobs, my partner, will be giving the presentation on the medicine, but first I'm going to give a brief introduction. Życie is a Ukrainian word for life. Our company is on a journey of discovery. Jack and I have been working for over 20 years on a series of medical indications that we believe will defeat the major causes of death in the world today. We believe that our treatment can defer the death from heart disease, from stroke, from Parkinson's, from Alzheimer's, and more. This is a journey of discovery. Jack is going to be explaining to you the medical theory. He's going to show you the medical science. He's going to then show you clinical data where we have succeeded in human clinical trials. We have 19 medical indications that we're working on. If you see a particular indication that interests you, Alzheimer's, peripheral artery disease, you can contact us and we'll give you a white paper on the issue. And for those of you that have a specific disease, Jack has already done presentations like in Alzheimer's, enhanced stroke recovery, peripheral artery disease, and you can go to YouTube and look up Angiogenesis Medicine, and we have 9 different presentations that Jack has done in the last 2 months there that can give you the science and all the information. But this is our first one on additional quality years of life, because we've evolved to understanding that we may be able to do a lot to defer death and give you another 20, 30, 40 years of quality life. Now, I'm 72 years old. I would like another 40 years of healthy life. I want more than that if I get there, believe me. But for right now, 40 looks pretty good. So at this point in time, I'd like to introduce my partner, Dr. Jack Jacobs. Dr. Jacobs is a PhD in molecular chemistry and biology from Washington University in St. Louis, commonly known as WashU. Jack is a brilliant PhD. He worked at Hitachi Pharmaceuticals. He went to Merck Pharmaceuticals where he set up their microbiology labs. And Jack joined me 22 years ago when he and I both looked better. We've been working on this for the last 20 years. He and I have expended in excess of $140 million. And we believe that we are on the verge of revealing to the world the biggest medical breakthrough in the last 100 years. Now, before I turn it over to Jack, I'd like to do a small quiz with you, ladies and gentlemen. And the quiz is very simple. 100 years ago, in 1918, what was the average life expectancy of a man in the United States? Take your time, think about your answer. Just like Jeopardy, folks, think about your answer. What was the average life expectancy in 1918? Answer, 38. Half of all men were dead by the age of 38. Now, the reason that's important is that if half of us were dead at 38 and none of us made it past 60, this would be a very lonely webinar. However, with the discovery of antimicrobials, starting with penicillin, the war with the bacteria switched from people dying of bacterial infections, tuberculosis, whooping cough, cholera, pneumonia, syphilis, all of these bacterial diseases, which used to claim the lives of half of the people, have now been dramatically reduced. Now the diseases that are killing us are diseases of aging. And we believe most of those diseases of aging are related to insufficient blood flow to a tissue or organ. Dr. Jacobs is going to talk to you about the next 100 years. I'm planning on being there. I hope you are too. Dr. Jacobs, please take over.
Thank you, Dan, for that nice introduction. Actually, Dan will be joining us at the end of the talk during the question and answer period, and also we'll have some closing comments. Okay, so my talk is going to be based into two different categories. As Dan mentioned, we're developing a drug for FDA. This is FDA clinical trials, FDA approval. And we're using it to treat diseases that cause more than 50% of deaths in the world. And I'll show you data from clinical trials on reversing heart disease. We'll talk about diabetic foot ulcers. And we've got some really nice data on neurodegenerative diseases, such as Parkinson's disease. And we're ready to start clinical trial in Parkinson's disease. So once we get people through these diseases, as Dan mentioned, we can use the same drug, which I'll talk about in a minute, how it acts on to restore our blood vessels, to regenerate blood perfusion to the brain, heart, muscles in healthy people, in normal, healthy 65, 70-year-olds. And we're going to talk about the lining of the blood vessels. They're lined with cells called endothelial cells, and how our drug can repair those, regenerate them, and really bring our blood vessels back to a healthier state. Okay, we all have almost 60,000 miles of blood vessels in our bodies, enough to wrap almost 3 times around the world. And these are all formed in utero or as we're growing by a process of angiogenesis, which is simply the growth of new blood vessels. Now we know when these blood vessels get blocked in the heart with coronary artery disease, we can get heart disease, heart attacks. These vessels get blocked, leading to the brain. We can get stroke. But also I'll show you at the end of the talk, as we age, our blood vessels become leaky and they don't function as well. And we'll talk about how that affects various parts of the body, but particularly the brain. Now, Dan mentioned we're working on 19 separate medical indications, but in fact, over 75 human diseases are a result of impaired blood flow to a tissue or organ. And so one way to get better blood flow, let's say to the heart, if someone has heart disease, is to grow new blood vessels in the heart, get more pipes delivering more blood, more perfusion into the heart, to that ischemic, starving heart muscle. And I'll show you an actual video of someone's heart who got our drug and where we grew new blood vessels after about a 3-month period in an FDA authorized clinical trial. But growing blood vessels in the body, it's a process known as angiogenesis, and we use it all the time. And the drug we're developing is probably the most potent stimulator of this natural process. So let me talk a little bit about angiogenesis in our bodies. And let me give you an example. Here's a skateboarder. He fell off his skateboard, scraped his elbow there, and he's going to use angiogenesis to heal that skin tissue and all parts of the skin tissue. So let's take a closer look at his elbow there. We're going to go, here's his wounded area. This is a scab that's over his scrape. And if we look underneath that scab, here we see angiogenesis in action. Here are these tiny new blood vessels that have formed, stimulated by our drug that I'll talk about in a second. This new blood perfusion heals that skateboarder's elbow, new skin, new blood vessels, new nerves, sweat glands, hair follicles, whatever is needed to regenerate skin as an organ. Here's a drug we're developing, human FGF1. It's a natural product in our body. Again, one of the most potent stimulators of new blood vessel growth of angiogenesis. It's most abundant in our brains. And in fact, I was involved in purifying this protein, as Dan mentioned, at Washington University in St. Louis. We got it from cow brains. We don't obviously use cow FGF1 in our clinical trials. We have recombinant human FGF1, which we're using. So let's look how FGF1 stimulates angiogenesis. And that skateboarder's elbow, let's say, as soon as he scrapes himself, his body starts making, in that wounded area, FGF1, which are these little flying things. They migrate to the blood vessel here. We can see them binding to the FGF receptors. So this is a very important interaction. FGF1, the drug, binding to its receptor, more or less like a key going into lock. And that starts this whole process of new blood vessel growth. Now, 2 things are important here. You can see there are many more receptors on this side of the blood vessel than over on this side of the blood vessel. So this permits the new blood vessels forming to grow into the injured area where it's needed and not into healthy tissue over here. So when we injure our bodies, the FGF1 receptors get upregulated right where they're needed, oriented in the right way. And it's been shown that in injury, FGF1 receptors are upregulated about 100-fold over normal tissue. What happens there? We get stimulation of these endothelial cells, which form these little tubes. They get elongated, and they actually meet up with other tubes growing from the other side, and you get this nice, rich, new blood flow in this wounded area, which stimulates regeneration of that tissue. Now, I want to talk about 3 examples where we successfully used FGF1 to stimulate therapeutic angiogenesis. So when we cut ourselves, it's an acute situation, and the body can generally take care of that. But in these chronic diseases, such as diabetic foot ulcers, coronary artery disease, Parkinson's, there's a slow deterioration of the blood flow and blood supply, and the FGF1 system needs a boost. So we actually go in and introduce what we call therapeutic amounts of our growth factor. So let's look at some of the data coming out of FDA clinical trials with FGF1. I'm going to start down here at the bottom, and we'll work our way up through the body. We'll talk about diabetic foot ulcers first. 38 million Americans suffer from diabetes, and 5 to 7% of them will develop every year a diabetic foot ulcer. And these are very nasty chronic wounds, can be open for years. They're deep, and they often get infected. And once infected, can lead to gangrene and probably the most dreaded consequence of a diabetic foot ulcer, which is a lower leg amputation. In an FDA phase 2 trial, FGF1 was put right into the ulcer topically, and we looked at its healing ability over about a 5-month period. So let me show you the results of that clinical trial. Again, it was a 2B trial, and remarkably, 100% of the treated ulcers with FGF1 were closed by 4 months. So here we are at the beginning of the clinical trial. 2 groups of ulcers, ones that are going to be treated with FGF1 in blue. In red are placebo-treated ulcers, so no drug. They're all wide open here in the beginning of the trial. And then as we treat over time, 30, 40 days, 50 days, out to 130 days here, you can see all of the FGF1 treated ulcers completely closed. So that's a very pleasing result, where the placebo-treated ulcers, about 35% still remain open. That's a big deal. That's a very big deal. These ulcers can get infected, a lot of economic problems, misery, and so this is really a very attractive clinical development program, which we're going to be pushing ahead with a lot of vigor. Let me just show you what a closed ulcer looks like. Here we see before treatment with FGF1. This is the open ulcer, and then this is after about 4 weeks of treatment with FGF1 to completely close this ulcer. Okay, now let me move to a second medical indication, coronary artery disease. Here, completely different application of the drug. We're going to be injecting it through a small incision. Actually, we're using a catheter now, but I'm going to show you a clinical trial where the FGF1 was injected through a small incision in the chest wall and directly into the hearts of patients who were suffering from severe coronary artery disease. And these were people who may have had bypasses or stents in the past, but had really no options now. So this was really their only treatment option. And I know the results of this trial very well. I supervise it, and actually the results came out so nicely that ABC Nightly News went to our Cincinnati site and interviewed the cardiologist there and some of the patients who were treated. So rather than me talk about this, let me show you this ABC News clip. And I'm not going to talk over the news clip, but I want you to pay attention when Dr. Wagner points to the areas of new blood growth, new blood vessel growth in the patient's heart.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So that woman we saw there, Constance Donnelly, she came into this trial in a wheelchair. She was a cardiac cripple. You can see her there 3 months after the treatment. She's up and about. We had 3 dosing groups in this trial, and she actually was in the lowest dosing group. She got what we thought was 1/10 of the optimal dose of FGF1 into her heart. So she must have had tremendously upregulated FGF1 receptors. She was primed to react to our drug. Going to move now on and talk about Parkinson's disease. Again, work from a lot of different labs across the country and world have discovered that a number of neurodegenerative diseases such as Parkinson's, Alzheimer's, MS, appear to be initiated by a lack of blood flow to certain regions of the brain. And that lack of blood flow begins choking off neurons. And I'll talk about this example with Parkinson's, where we believe it's choking off the dopamine containing neurons, which are the ones affected in Parkinson's disease. And that's shown on the next slide here. We have billions and billions of neurons in our brains, but we only have 500,000 of these dopamine neurons, the ones that are become dysfunctional in Parkinson's. And they're contained in a very tiny region in our midbrain. And it's called the substantia nigra. That's because these neurons are dark, shown here. I'll show you some perfusion data, but lack of blood flow to this region has been shown to choke off these neurons and causing them to become dysfunctional. And we know with a normal neuron, we have normal movement, motor movement, but as these neurons become affected in Parkinson's, they release less dopamine, and we see the characteristic movement disorders in Parkinson's, the gait disturbances, the tremors. Let me show you some data we got in a monkey model of Parkinson's. This is the gold standard where drugs before they enter the clinic have to be tested. And we can give this little guy Parkinson's disease by injecting a toxin into his brain. And actually, there's a tragic story. This is a herbicide. It's used to kill weeds. And in the 80s, a marijuana grower in the Seattle region sprayed this toxin on his crops. Some teenagers broke in at night and stole the marijuana, smoked it, and unfortunately came down with irreversible Parkinson's disease. So that same toxin is used, injected directly into the brains of these monkeys. And then we measured their motor scores. So 5 is perfectly normal movement by the monkeys, and 1 is very abnormal movement. So just one injection of this toxin, they wait about 9 months. So here we have 2 groups of monkeys. At this point in time, we're displaying all the classical symptoms of Parkinson's, gait disturbances, they're jerking around. And one group gets treated in blue with FGF1, an IV infusion, and the second group gets a placebo dose. And you can see over the months, the FGF1 treated group actually begins reversing their motor disturbances. So here they're about 80% of normal. I think perhaps it will be treated longer. They might even go higher. The placebo-treated animals are doing very poorly. And the difference in motor scores between this and this is highly significant. So based on this, we appear to be reversing the motor dysfunction seen in Parkinson's in these animals. And we then wanted to look into the brain of these animals to see if we're actually regenerating the dopamine producing neurons that have become dysfunctional in this disease. And in fact, we are. We can look at the brains of these animals. We look right in that area of the brain where the dopamine neurons are, the substantia nigra region of the brain. They stain, new dopamine neurons stain brown. So we can see here in that region of the brain housing the dopamine neurons, we have this whole population of brand new dopamine producing neurons, which we feel is restoring the motor activities in these animals, where you don't see that in the placebo treated animals. So we think we're really attacking this root cause of Parkinson's. We're regenerating new dopamine cells in these monkeys, and we're restoring motor function to these monkeys. Now, one other aspect of these neurodegenerative diseases, seen in Alzheimer's, where you get accumulation of toxic plaques, these aggregated proteins in the brains of people with Parkinson's and also Alzheimer's. The same thing is seen in these monkeys. In the monkeys with Parkinson's disease, here's a toxic tangle of proteins right in that small area of the brain where the dopamine neurons are. This is in the substantia nigra region. This is a synuclein protein, so this is different than Alzheimer's, but again, this is toxic to the dopamine neuron. So it's kind of a double whammy. Less blood flow, this toxic protein accumulates, and we can see here in the treated animals, we've really cleared a lot of that aggregated toxic protein away. Not quite back to normal, but certainly better than this situation. So these two disease-modifying effects, regenerating the dopamine neurons, whisking away this toxic neuron, we believe lead to the restoration of motor function in these monkeys. And we're very excited and interested to get this into the clinic. And we've designed a clinical trial. 3 ascending doses of the drug will be studied, no placebo group. All patients will receive a 1-hour intravenous infusion of FGF1 5 days a week through a central line, a PICC line, so we don't have to be sticking them every day. And this will be the first proof of concept study with our drug in a brain disorder, and specifically in Parkinson's disease. We'll mainly be looking at safety, but also testing for any effectiveness. Can it restore motor function in patients with Parkinson's? As long as it's safe, we can tweak the trial. We can do an additional trial with larger doses of FGF1. We can give it for longer periods of time till we start seeing, hopefully, some efficacy in this trial. We submitted this trial to the FDA. They approved of the design, but since it was the first time FGF1 was being given for a long period of time in a brain disorder, they've asked for an additional animal study, a toxicity study, which we're doing. The same trial was submitted to the FDA in Mexico. COFEPRIS is the name of it. They liked it, didn't want any additional animal trials, and we're set to start there as soon as the pandemic lifts. And we also have clinical trials under review in Estonia, the Baltic country, which is an EU country, and also in Morocco. So we're trying hard to get a clinical trial going because we want to know as soon as possible if we can help people with Parkinson's disease. We're also developing drugs for these other 19 medical indications. You can go to our website, zgm.care. You can sign up for these to receive more information on these indications, learn when we're starting clinical trials. I talked about heart disease, but we also have a stroke recovery. And Dan mentioned peripheral artery disease, PAD. This affects the legs, blockages of the legs. Wound healing disorders, these are all pretty much topical applications on diabetic foot ulcers. We have clinical trial data on venous ulcers. These are very large, painful ulcers in people's legs. Bed sores are a real problem in nursing homes. Surgical wounds, FGF1, it's noticed when it closes a wound, does so with less scarring. So there might be some cosmetic applications as well. Burns, and then not really a wound healing, but something very interesting. We kind of through serendipity in animals, if you inject FGF1 subcutaneously, you get this luxuriant hair growth coming out. So it's possible this could be also a treatment for male pattern baldness. All of these brain disorders, this is not our work, this is work coming from a lot of different medical centers around the world, are initiated by a lack of blood flow. When I talk about Parkinson's disease, the same with ALS or Lou Gehrig's disease. We've been approved to start an ALS trial in Mexico. We're going to look at Alzheimer's disease, chronic depression. FGF1 levels are severely depressed in the brains. I did a webinar on that if you want to look at that. MS, we have an MS webinar given by Vika Montano. Traumatic brain injury, PTSD. CTE is this disorder that boxers and NFL football players can come down with after repeated concussions. Vascular dementia, MSA, which is a kind of aggressive form of Parkinson's disease. So all these are diseases that we think we can treat, and we're going to test. But I want to move now on to the second aspect of our drug, and that its ability to repair and restore the deteriorating vascular system. And we think this is very important for life extension. With better blood perfusion to the brain, heart, muscles, we think we can counteract the gradual decline of our bodies as we age. At the very end of this talk, I'm going to show you a video out of Columbia University Medical Center, where they think the reasons our brains decline as we age, and this is in healthy aging people, is because of a lack of blood flow. But let's look at vascular dysfunction. New research indicates that as we age, our blood vessels don't do as well. They become leaky. They leak stuff out that should be within this blood vessel, and sometimes what they leak is not all that good for the surrounding nerves or whatever is around there. So let's look at this a bit more carefully. The vessels, here are the endothelial cells. That's a very important cellular component right here. These are healthy endothelial cells that as we age become leaky. And I'm going to show you an example of this. It's called endothelial cell dysfunction. And this is occurring both in diseases, this is seen in MS, but also as we age. Here's the normal endothelial cell layer. This is inside our blood vessels, keeps the blood vessel intact. Then either through disease or aging, we see this primary endothelial cell dysfunction. You can see these are not as nice looking endothelial cells lining the blood vessels. They get bumpy, they become leaky. And in an animal model, I'm going to show you in a second, we can heal that. This is a diagram of what's going on where dysfunctional blood vessels can leak immune cells and toxic proteins leading to damaged nerves. This is thought to happen in MS, but also we believe is happening as we age normally. Healthy blood vessels in the healthy brain, optimal blood flow, healthy neurons. As we age or through the disease process, we develop this endothelial cell dysfunction. And that leads to things leaking out of our blood vessels. We have one, reduced blood flow, but two, we have these immune cells. We know in MS it's thought of as an autoimmune disease. Well, in fact, yes, these immune cells are leaking out and attacking these neurons. Toxic proteins, here you can just see here fibrinogen. There's no reason that should be anywhere near a nerve cell. That's a clotting, blood clotting protein. It's a sticky protein. And it'll actually coat these neurons and make them more immunogenic, more susceptible to attack by immune cells. So we think FGF1, if administered here, can help heal these dysfunctional blood vessels. And in fact, in an animal model, we can restore and heal damaged endothelial cells in these capillaries. So this is in an animal model of MS, multiple sclerosis actually. Here's this endothelial cell dysfunction, and then we treat with FGF1, and over a period of days here, by day 30 here, you can see a nice remodeling, regeneration of the inside of the blood vessel in these animals, no leakage out of these vessels at day 30. And you can see with histology, you can actually look at the endothelial cells. Here's early on leaking stuff out, dysfunctional. We heal them with FGF1 here. They're completely healed, very little leakage from these blood vessels. So we really are excited about trying this in models of aging, and think this has a real shot to be something to look at to extend our blood vessels and then extending our lives. Let me show you now this Columbia University Medical School, very prestigious medical school in New York City. They published a paper in a very prestigious journal, Cell, and then Time Health actually did a video on this. I'll show you the video about why our brains perform worse as we age. And this is looking at healthy people. And I want to give you the punchline before I show you the video, because it's important for what we're doing. They're going to talk about stem cells. And I know people associated with red know about stem cells. In this video, they're going to talk about how 90-year-olds have as many stem cells as 10-year-olds. But what's lacking is adequate blood perfusion in the 90-year-olds to keep these active, okay? Stem cells are very important. For example, when I just showed you in Parkinson's, we know a lack of blood flow is affecting the dopamine neurons, the mature neurons, but probably lack of blood flow is also affecting these neural stem cells, which in healthy individuals can go on to form new dopamine neurons. So it's kind of, again, like a double whammy. You're making the existing neurons dysfunctional, and you're not stimulating these neural stem cells to actually be able to differentiate into healthy neurons. I'm going to show you this, the video now, and I won't talk over it, but I think you'll agree that this supports what we think about the importance of blood flow in the brain. So we agree completely with this video. And we think we have a drug that can restore blood flow to the aging brain by healing endothelial cell dysfunction, leakage. And so we're excited to get into that area as well as using it to heal these diseases caused by a lack of blood flow to the brain and other tissue. So again, here's our clinical trial website, zgm.care. You can go there to sign up, learn more about our clinical trials. And I'm happy now to answer any questions you may have. And I'll invite Dan Montano back. There he is.
Hello, Jack. Excellent, as always. By the way,
you.
Jack, I think you need to put the slide back in that has the artist's rendition of the FGF1 occupying the receptors.
Yeah, okay.
That may help us explain some things, because we got a lot of questions here from a lot of people.
Okay.
So let me start off with the one that we always get, the biggest one. A lot of people want to know, does FGF1 cause cancer or make cancer spread?
Yeah, that's a very good question, and it comes up. FGF1 is a very potent growth factor. And because of that, our lovely FDA made us test it in animals. So at 100-fold the doses that we're using in our clinical trial and given for longer periods of times, we saw no induction of cancer in the animals. Second, we've treated probably close to 90 people in the various clinical trials, no hint of cancer. But just to be extra careful, one of the exclusion criteria, that means something, if you have, you can't get into the trial. So cancer, active cancer is an exclusion criteria. So nobody with cancer can get into the clinical trials, just to be on the safe side. But I have never seen FGF1 cause any type of tumor or cancer in an animal or a patient.
Okay, Jack, next one, is FGF1 similar to human growth hormone?
Again, they're both growth factors, but they're different. So they have different receptors. FGF1 mainly acts on nerves and blood cells. growth hormone stimulates what's called a somatomedin, and that acts on the bones and tissues to stimulate growth. And also muscle tone. As you get older, people take human growth hormone for muscle tone as well, but they have completely different structures.
Okay. Jack, why don't blood vessels start growing all over the body when FGF1 is given?
Good question. I kind of alluded to that, but only in damaged tissues do you get that tremendous upregulation of the FGF1 receptors. So I've talked about 100-fold upregulation, so it kind of concentrates our drug there. If you put FGF1 into normal tissue, into normal heart tissue, basically nothing happens. No blood vessels grow. So it's only in starving tissue where the blood supply needs a boost do you get the FGF1 acting.
Jack, we have a question. PICC line, would that be to the brain, into the brain?
No, the PICC line goes up through the arm and into the vena cava, which is the major vessel coming out of the heart. But we believe that would get the FGF1 to the brain very quickly versus putting it into a artery in the arm. And the second thing we do, the person's going to be treated 5 days a week for 4 weeks. We do a lot of blood draws, so this can be really cuts down on harassment of the patient by having a PICC line in there for the duration of the clinical trial, for the 4 weeks they'll be treated.
Jack, one just came in that I'm going to take out of order, but would basal cell cancers disqualify someone for this trial? And I think what trial are we referring to, right?
Actually, no, that's the only, that's the one cancer we allow. The FDA allowed us that a basal cell carcinoma and is allowed in this trial. So that is the one that we do allow.
First you tell us no, then you tell us yes, Jack.
Well, there's a lot of worse cancers than basal cell carcinoma.
So next question, Jack.
Okay.
For Parkinson's, will FGF1 mediate a generic duplication of the SNCA gene for the Parkinson sufferers, or do they need to regularly come in for treatment to counteract the alpha-synuclein problem?
Okay.
I think I'm an accountant, huh?
Yeah, I'm not, I know the alpha-synuclein problem. I'm not aware of drugs that treat that. I know they have experimental drugs, antibodies, but the patients who come into our trial will keep taking their Parkinson's medications, all the dopamine, products.
Jack,
yep.
Jack, the Jewish population, I think it's Ashkenazis have a propensity to Parkinson's, and a lot of people think it's a gene. This may be referring to if you have that gene, and that gene is generating the nuclear problem, can FGF1 address it? I think that's a better framework for you.
Yeah, that's a good question. So at first, we're not looking at the genetic forms of Parkinson, which is about, I think, 10% of Parkinson's is genetic. We're looking at what's called idiopathic, so spontaneous Parkinson's, but I see no reason not to look at the genetic forms if, again, that could be helped by a boost in blood flow, but at first, we're not going to be looking at that form of Parkinson's.
Okay, Jack, I got to take this one out of order here. This one came in and I love it. I am a surgeon living in the US, but I have clinical access across the border. Is it available for use in other countries like Mexico? I'm thinking about laparoscopic injection in some patients.
Are you going to take that?
No, you take it. I don't like the tricky ones.
Yeah, no. If you can practice medicine in Mexico and we get approved in Mexico, we would invite you to our clinics, or you can open your own clinic and treat patients in Mexico through laparoscopic or whatever way is deemed appropriate, depending on what disease you're treating. Obviously, if it's a diabetic foot ulcer, it would be topical, but there could be other indications like the heart where you would do a laparoscopic type of procedure.
And I want to emphasize, we believe that there's a whole business industry around our drug getting acceptance and the rehabilitation, and for people, if you've had a stroke or if somebody's had peripheral artery disease, we believe that we can fix the root problem. We can fix the fundamental problem grow new blood vessels, reestablish perfusion to neurons. But people still have to be rehabilitated. Those nerves have to be taught. So we're always looking for business ideas. Okay, another question for you, Jack. What are the side effects that have been seen in the clinical trials?
Okay, that's always a good question. To date, there have been no serious adverse events attributed to our drug, which is really a great result. And again, with the animal studies, if you go to 100-fold higher levels, the FDA wants you to see, pushes you to see adverse events. And there we see some organs start to be affected. The thyroid begins to grow, and it's reversible. If you stop the drug, it'll go back. But in the clinical trials, we have not seen any adverse events that can be directly attributed to FGF1. We look for tumor markers. We look for in the eye, at the retina. We don't want any neovascularization, blood vessels growing in the eye, and we have not seen any of that to date.
Jack, I'm going to take another question out of order here because I love this question. One of the viewers looked at me and wants to know the following. Down the road, could there be a cosmetic purpose for this drug? I think they are assuming I need it.
Yeah, I think, Dan, you could use it, probably we all could, but as I mentioned, in animal models, in surgical wounds, made with scalpels, FGF1 applied to those surgical wounds heal with less scarring, less scar tissue. So that I think could be a very important medical indication. It's one that's on our list.
Okay. Now, Jack, I'm going to ask this question, which has not been asked formally. One of the people who is presenting, I've talked to that person on the phone in Florida. Her sister was in an automobile accident and had a traumatic brain injury. She had severe trauma. And of course, I talked about, we're looking at traumatic brain injury. And I know that's not the topic here, and I know it's a totally different presentation and different slides. But I really like this person, and I think she would appreciate it if you were able to comment on the idea of somebody's had a traumatic brain injury. What's the effect that we've seen with the vascularization, and what do we think could modify that, please?
Sure, yeah. So we know that after a traumatic brain injury, there's damage to the vasculature. There's animal data, good animal data in animal models of vascular traumatic brain injury that FGF1 given intravenously can actually heal the lesions in these animals. So this is definitely on our list. It's similar to, we think, to stroke recovery, that we can infuse FGF1, we can recover the vasculature, which can lead hopefully to repopulation of the nerves. So it's definitely something that we're going to try and have hope.
Okay, the next question is, do you feel your drug can rejuvenate the blood vessels as we age? And I'd like to make a comment. I'm 72. As soon as we get demonstration that this molecule is safe at dosing level, I'm going to call it 3, that it's safe there, then that's the point I'm going to try it. Because if it's safe and it does no damage, as we've seen in other organs and other tissues, I'm going to go in and make sure my brain doesn't shrink, that I don't lose my mental capabilities as I get older. I would rather not lose blood vessels, neurons in my brain, rather than try to repair it. So Jack, the question is, do you feel your drug could rejuvenate my brain and keep me going?
Yeah, well, I think, I showed you that one slide where we could repair those dysfunctional endothelial cells in the blood vessels of those animals. We'll be doing clinical trials in diseases that have primary endothelial cell dysfunction. And if we see healing of those vessels, I see no reason not to try it in an aging population and to look at very carefully defined endpoints, such as cognitive decline, things of that nature. But I agree, if it's safe, why not try it?
Jack, here is another question. What about endothelial cells in the gut? Would it work for leaky gut or oral cavity?
Wow, it's a good question.
This person's tough on us, Jack.
Yeah, there's some, we've looked at vascular diseases of the intestines where you actually get blockages in the blood vessels supplying the intestines, and that causes disease. And I don't know if that's what you're referring to, but that also can lead to leakages. But yeah, we've looked at gut diseases which are characterized by blockages of the blood vessels, and it's possible by injecting it into the local regions in the gut, we could overcome that problem.
I will also comment that I had a conversation last week with a very successful doctor who owns several hospitals in the West. And he wanted us to go over the endothelial cell dysfunction, and he wanted to see our evidence. And his response at the end of that was, this is bigger than you realize. This can help in the guts. It can help in the liver. It can help in the kidneys. This is bigger than you realize. You have no idea how important this is. And we got all excited about it because it appears that endothelial cell dysfunction may be taking place throughout the body. And sometimes your body repairs it naturally. Maybe other things you do can enhance it. But if FGF1 proves to be safe and can rejuvenate that tissue, we don't see any downside. And so we're very excited about it. Now I have another one here from my favorite person, and she knows who she is. PEMF, like Beamer, if used with FGF1, might increase positive results.
What was the first thing? What's that?
That goes to you.
Okay, could you repeat the first part of that? What was the compound?
It's not a compound, it's the electronic mat that vibrates. Remember Beamer?
Oh, right. Yeah.
Everybody at Rad has a Beamer except you and I. I thought there was a car.
I'm sorry.
Somebody asked me if I had a Beamer, and I said, no, I have a Ford.
No, I like those mats. Those mats increase blood perfusion. So I think, ours is a therapeutic. You have to inject it and have to get it through a prescription. It may be longer acting, might grow new blood vessels. But the two in concert, I think, is worth looking at because those mats do increase blood perfusion. Like exercising can stimulate angiogenesis. So I think it's a good idea.
All right, Jack, we have one. How long will the new rejuvenated endothelial cells live?
Yeah, that's a good question. We don't know the answer to that. We know in our heart studies, the blood vessels we grew in the patients injected, we looked at them 3 years later. They were still there, the ones we'd grown 3 years before. So obviously those endothelial cells are still doing well 3 years later. We hope it will be a durable response that will last for years, but we don't know the answer to that at this point in time.
Well, one thing too, I think all of us recognize is that the cells in our body are constantly regenerating. So the lining of our small intestine rejuvenates about every 6 days. The skin rejuvenates around every 20 days. The fact that I have good skin doesn't mean it's the same cell that was there 13 years ago. It means it's rejuvenated and modeled in a proper way that stays there. So the FGF1 may fix the problem. It may initiate the problem. It may change the condition, and the continual reproduction, the continual renewal may go on. We don't know if it's going to be 3 months or 30 years. What we do know from the heart trial, when we injected the drug in the heart trial, we had patients that were on death's doors, and those patients were treated, and 10 years later we had a reunion for them. When we had the reunion, 9 out of 10 had changed their lifestyle. They'd lost weight, they started exercising, they quit smoking, blah, 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 and their hearts looked fine. And one guy, when he got the treatment and his heart was regenerated and his pain went away, he went back to smoking, eating, chasing, and whatever, and he was having pain again. So we think there's a combination here. We can regenerate things, but it's up to you to make sure that you don't re-damage them. I have one more question, I think, Jack. Would this be an appropriate strategy to increase cardiac blood flow impeded by calcium score of 660 and obviously blocking some amount of blood flow in the heart? So this is a calcium blockage question.
Absolutely. The calcium scores kind of measure atherosclerosis, and we measured also with our angiograms, but absolutely, this is what this drug would do. It would grow new blood vessels around blocked arteries and increase blood perfusion to someone's heart who's starving, who needs more blood. So that's exactly what we showed in that clinical trial. what our drug can do.
And I want to emphasize, in the heart trial, we did not repair the closed arteries or arterioles or capillaries. We grew new ones around the problem. So if there's calcium blocking or narrowing, we're not going to address the calcium. We're going to replumb around it. And this is important because what a lot of people don't realize who have heart disease, when they go in there and they open that up with a stent and they rupture with an angioplasty, they rupture the blockage. That gunk goes somewhere, people. It doesn't disappear just because they stuck a stent in there and expanded it. It goes somewhere. And that disease material goes throughout the body. And the heart or the brain is one of the primary filters and receptors of that. A lot of people go and have a stent procedure and they basically create vascular problems in their brain. And this can be documented. In our case, we don't disrupt those beds. We don't disrupt the blockage. We go around it. And that we think is the safest way to go. We have one last question. Where does the name Zhicha come from? Vika, come here. Nina, this is for you. This is my wife, Victoria. Tell them where Zhicha comes from.
Zhicha is Ukrainian name for life. And the symbol is actually also means life by itself.
Zhicha means life. Genesis means a new beginning. So Angiogenesis means life's new beginning. And I want to thank all of you. And we hope that you will go to the YouTube. We hope you'll send us questions. We hope you will follow our journey. I had somebody who asked me last week, said, I think you're going to fail. I think you're an idiot. I think this is bull. They had another word attached to it, but I'm not going to use it. And I said, fine, then follow us. Come and watch if we succeed or fail. Jack and I've got 22 years in this, $140 million. I got 26 million of mine in. The issue is we believe we've got something here. We think you should take a look, follow us, laugh at us if you want, but people, we've grown blood vessels in the heart. That's been evidenced 4 different human trials. We've healed diabetic foot ulcers 100% of the time. That's been proven in phase 2 trials. In the brain, we have 2 big issues. Is the molecule safe? And does a molecule provide benefit? I think we're going to know that in the next 12 months. You want to know if we fail, hey, we fail. But if we make it, this could change a lot of people's lives. This is Dan Montano and Jack Jacobs. Jack's a smart one. I'm the cute one, saying thank you and have a good day. Bye-bye.
Thank you. Bye-bye.